Hello, this is microbiology. Today we'll be discussing the remaining part of our topic, that is culture media. Simple media. Simple media, the media that support the growth of non-fastidious microbe, and it is primarily used for the isolation of microorganisms. Example are neutron broth and neutron agar. So it's mean that media doesn't require any special nutrient. And the primary use of this media is for the isolation of microorganisms. So which bacteria can grow on this media? It may be Staphylococcus species and Enterobacteriaceae species. Second one is enriched media. So the enriched media is basically prepared by adding additional and some special substances like blood, serum, egg into this sample media or based media, so it will become enriched media. This media is usually used for posterior microorganisms that require some additional nutrient and special substances for their growth. Example of these media are chocolate agar, blood agar, and low blood serum slope. Chocolate media is used to grow Neisseria gonorrhea, while blood agar, which is prepared by adding 5 to 10% blood by volume to blood agar base, is used to identify hemolytic bacteria. And we already discusses how how these media are prepared. Chocolate agar, blood agar, and low blood serum slope. We already discussed in a previous lecture. The third one is selective media. This media allow the growth of certain microbe while inhibiting the growth of other. So it means this media is basically it enhance the growth of those bacteria which we required, and while inhibiting the growth of other. It may be normal flora or other bacterial species. It is basically agar-based medium. It means that are solid medium. It's used to isolate the microorganism in lab. So how we make this media to selective? By adding certain substances like antibiotic, dyes, bile salt, or by pH adjustment. Example of this media are thiamartin agar. It contains antibiotics, vancomycin, colistin, and nystatin. This antibiotic is used for the selective growth of Neisseria gonorrhea while inhibiting the growth of other bacteria. And the second example is McConkie agar. It contains bile salt. It is a selective substance. It's a selective molecule. It basically enhance the growth of Enterobacteriaceae species while inhibiting the other bacterial species. So the third example is Livingston Johnson medium that is used for Mycobacterium tuberculosis, and the selective growth substances is malachite green that enhance the growth of Mycobacterium tuberculosis while inhibiting the other bacterial species. So the fourth one is in enrichment media. The maximum principle and procedure is same of this media like selective media, but the selective media is solid media, and the enrichment media is a liquid type media. So the main use of this media is used for to isolate fecal and soil microorganism. Example are selenite F broth, which is used to isolate Salmonella typhi from a fecal sample. Tetrathionate broth and alkaline peptone water. So alkaline peptone water is used for Vibrio and cholera, and tetrathionate broth is used for Shigella species. The fifth one is the differential or indicator media. It contains certain indicator like dyes or metabolic substrate in the medium composition which give different color to the colonies of different microbial species when they utilize or react with this component. So here in this medium is used different indicator that is maybe dye or like different substrate. So when this dyes, when the bacteria react, when it will produce certain color, different color. By this color we can identify these bacteria. So the important advantage of this media is that it allows the growth of more than one microorganism. So within this media, we identify the bacteria on the basis of their different color colonies they are produced, and we can easily identify this bacteria when the bacteria react with this certain indicator that is used in this media, and as a result it will produce certain color, and the color may be neutral red, phenol red, methylene blue. Example are mannitol salt agar, McConkey agar, blood agar. So the mannitol salt agar is used to identify the Staphylococcus aureus, because Staphylococcus aureus is mannitol fermenter. It will produce yellow colonies in the media, while the rest of other step species they remain colorless. The second one is the Meccan agar. The Meccan agar is used for the fermentation studies. That is basically used for the lactose fermenting bacteria. Lactose fermenting bacteria are E. coli, Klebsiella. They produce pink colonies on Meccan agar. While non-lactose fermenting bacteria are Salmonella Shigella. They produce colorless colonies on Meccan agar. So the third one is blood agar. It is used basically to differentiate between the different streptococcal species like alpha hemolytic streptococci, beta hemolytic streptococci, and gamma hemolytic streptococci. So the alpha hemolytic streptococci, the main example of alpha hemolytic streptococci is Streptococcus pneumonia. So if we grow the Streptococcus pneumonia on blood agar, it will partially hemolyze the blood cell in a culture medium because it produces an enzyme hemolysil. So it's a basic characteristic of this bacteria. We can easily identify these bacteria by their hemolysis, on the basis of hemolysis. So as a result, their colony will be greenish. While in beta hemolytic streptococci species, that is Streptococcus pyogenes, it lead to a complete hemolysis of RBCs, so it will be a clear colonies affair. So here we can see a clear hemolysis around the colonies. And third one is the gamma hemolytic streptococci, that is Enterococcus speculus, which can lead to hemolysis of RBC. So there will be no color found in the media. The last one is transport media. These are semi-solid or solid media. They contain ingredients to promote the survival of aerobic and anaerobic pathogen and prevent the growth of common cells or normal flora when a specimen cannot be immediately inoculated. So the example of transport media is that Kerry Blair medium for preserving enteric pathogen, and MS transport medium is used for ensuring the viability of gonococci. So here is the one main point is that the solid media we will be prepared in a culture plate, while the liquid media will be prepared in a tube, in a test tube, or in a conical tube. If you like this video, kindly share and follow my Facebook page. Thank you for watching.